retinitis pigmentosa. We know that mutations in one of 60 genes at this day, and actually every month we will find for sure one more gene, mutations in one of 60 genes can cause RP. We know that these 60 genes contain more than 600 building blocks, or these exons. To do standard sequencing using Sanger sequencing will cost about 10,000 euros. So you understand that this is for a routine diagnostic lab is a lot of money, so we need to find other techniques to find these mutations. So a new technique has come into our field, which has brought a revolution in our field, genetics, but also in biology in general. Because the technique initially was designed to sequence the complete genomes, all the chromosomes of all species that we know on Earth. And that's where it's mainly used for, to sequence complete genomes of all kinds of animals and plants. If we compare this new technology to the old technology, we can say that it's about 1,000 times more sequences can be generated using this technique. It's 100 times faster than Sanger sequencing, and it's about 50 times cheaper. So we can generate much more data with this technology. So let's have a look at this technology and how it came about. Just to give you the perspective, in 2002, about 10 years ago, there was, from the community of genetics, there was a press conference in which they claimed that they had actually sequenced the complete human genome, so all the 23 sets of chromosomes of a human being, using Sanger sequencing, and the costs were 3 billion, that's a 3 with 9 zeros, 3 billion euros, and it took hundreds of researchers 10 years to accomplish this. Now we are 10 years later, and the DNA sequence of one person can now be determined using this new technique, next generation sequencing, for 30,000 euros in 1 week. So that's 100,000 times cheaper if you compare the numbers. And what we are doing in a daily practice now, that we do not sequence the entire sequence of a human being, but we select those parts which encode for the proteins, which we call the exons, the building blocks. And those, let's say, building blocks of all the 20,000 genes that are around can be analyzed for 2,000 euros of one person in 1 week. So we can do much more than we could in the past. So in Nijmegen, where I am doing my research, we did a test of, let's say, the known retinal disease genes, I call it the RD gene package, that contains 150 genes. And we have done a study in the last 1.5 years to test all the known genes in a certain number of patients. So we specifically looked at 60 of these 150 genes to test patients with RP. So we analyzed 100 individuals that either had sporadic or recessive RP. And we analyzed all these genes as one gene package. We can find the causative mutations in about 55% of the cases. So what are the advantages and disadvantages? The one small disadvantage is, in this gene package, we cannot detect mutations in the X-linked RPGR gene. There's a technical reason for this. The second advantage is that it will not find mutations in genes that we have not yet uncovered. So we now know 60 RP genes, but we also know there must be at least the same number still out there that we haven't recognized. And using the gene package approach, we cannot find variants in new genes. So if we now look at all the genes, all the 20,000 genes of the human, advantage is that now we can find all the variants. Disadvantage, again, because it's the same technical limitation, we cannot find the mutation in this X-linked RPGR gene, in a part of this. So the other disadvantage is that we also will find some variants which have no meaning for the eye, but which could have a meaning for that individual for other tissues, like the brain. We have no idea at this moment exactly how frequent we will find these chance variants, but there will be some to be found. And it's probably less than 1% of the cases, but in those cases we will find genetic variants that predispose individuals for other diseases, other than RP. So we have to be very considerate of this fact, and so we have to discuss with each person that is tested using this new technology that he or she can be informed on this kind of results. So in Nijmegen we have established an expert committee that looks at all the variants that are found which could be important for the patient. And if we consider as an expert committee that this finding is important for the person for his own his own healthcare, then this will be reported back to the clinician that's involved with this patient, and he or she will decide how to communicate this back to the person. So when can we be sure that we have found another RP gene? First of all, we need to find a really severe mutation which disrupts the function of the protein. The protein also must be expressed in the eye, otherwise, why would it make sense? And we want to find mutations in other persons with the same eye condition. And the last question that just came from the last talk from the lady here in the audience, do we collaborate? Yes, we collaborate. And in the European Retinal Disease Consortium, as one example of many, we share data between 9 groups and in 8 countries, and we do this in an immediate way. So we are really very fast in communicating between each other. We have to do it because diseases that we are studying are so rare that if we find something in one family in our own cohort, it's very difficult to find a second case in our own small cohort. So we need to collaborate to look in a larger group of patients, and in the European Retinal Disease Consortium, we together look at 6,000 families. And then we will find a few other families with mutations in the same gene. For us, this is a prerequisite to be able to publish it. So there is an absolute need to collaborate. So when to use next-generation sequencing? Well, importantly, if a clinician comes across a disease which he or she has never seen, which is different from the already established diagnosis, then to do an unbiased analysis, next-generation sequencing will give you results of all the genes. So that's for sure a group that we want to test. And of course, it's important to look at all the dominant and recessive eye diseases for which several genes are involved, such as dominant RP or Cohen-Rod dystrophy. In persons with isolated RP, the inheritance model can be threefold. It can be recessive, and from our data, this is about 80% of the cases. It can be X-linked, again, about 10% of the male cases, but probably also a large proportion of female cases. And we also are finding mutations in dominant genes. And in this case, these are mostly novel mutations that we do not find in the somatic tissue, in the blood cells of the parents of the person with RP, but we do find a mutation in the lymphoblast DNA of the patient. So this is important to realize because the recurrence risk, so the children of affected persons, the risk to get RP are very different depending on the inheritance model. So if the inheritance model is, as was shown in the first presentation, recessive, then for the children to get the same disease, the risk is less than 1%. If there's dominant inheritance, the risk is 50%. And if we're talking about X-linked mutation in affected males, then there's almost, well, I could say there's no risk for male children to get the disease, because they inherit the X chromosome from the mother and not from the father. There's a small risk for female children, and the reason is that some females that carry one mutation in one of the 2 copies also will get RP. But more importantly, the sons of these female children, they have a 50% chance themselves to also inherit the disease. So clearly, for isolated cases, it's important to know genetic defect. So the take-home messages I want to give you today is that, first of all, Sanger sequencing is effective in eye diseases in which one or a few genes can be mutated. Next-generation sequencing is effective in eye diseases in which many genes can be mutated. Thirdly, next-generation sequencing of 150 genes reveals mutations in 55% of persons with RP. 4, in 10% of isolated male cases with RP, we find mutations in X-linked genes. And finally, in 10% of isolated cases with RP, new mutations are found in dominant RP genes, which I think is quite surprising. So I would like to acknowledge and illustrate, again, the question that was put forward, that we have a wonderful collaboration between these people on this slide, but I can assure you we have collaborations with all colleagues in the world. And I think this is one of the hallmarks of our field. So feel assured about this. And then I would like to thank also my people in my groups, actually a couple of groups in Nijmegen, that enabled me to discuss these findings with all of you. And I'm happy to take some questions from you. Thank you very much.